Good evening. My name is Alexandra Grajales, and it's my pleasure to welcome you here tonight. We would like to begin this event with a land acknowledgement. Iowa State University is located on the ancestral lands and territory of the Bakotsi, or Iowa Nation. The United States obtained the lands from the Meskwaki and Sauk nations in the Treaty of 1842. We wish to recognize our obligations to this land and to the people who took care of it, as well as the 17,000 Native people who live in Iowa today. We would like to thank the co-sponsors of tonight's talk, the College of Liberal Arts and Sciences and the Committee on Lectures, which is funded by the student government. A few housekeeping items. There will be an opportunity to ask the speaker questions. Please note that there are microphones set up on the side aisles for you to use, and please do not use flash if you are taking pictures. If you leave before the Q&A is finished, please exit quietly and refrain from talking until you have left the room. A lectures program student worker will be in the hall outside the room to scan your student IDs if you're here for class credits. Card scanning will not start until after the lecture is finished. Finally, I'd like to make a brief announcement on an upcoming event. On Monday, November 13th, University of Michigan Professor of Law Aaron Berzanowski will present his lecture, The Right to Repair, Reclaiming the Things We Own. This lecture will begin at 7 p.m. here in the Great Hall. All lectures are free and open to the public, and a full schedule of upcoming lectures is available on the lectures program website. Next, I'd like to welcome the Dean of the College of Liberal Arts and Sciences, Bjarne Schmidtmann, to the stage.
Welcome, and thank you all for joining us for the Fall 2023 College of Liberal Arts and Sciences Dean's Lecture. As you just heard, I'm the Dean of the College, and it's my pleasure to be your host for tonight's event. This evening's lecture is the 35th in this lecture series, which highlights faculty excellence and achievement in teaching, research, and outreach in the College of Liberal Arts and Sciences. Exceptional members of the LAS faculty are invited to present lectures from their own diverse areas of scholarly expertise. They are faculty members who help solve global challenges, connect the world with art and culture, and who push the boundaries of educational and intellectual dialogue. Our topics have ranged from new approaches to the development of treatments for specific types of cancers, to the war in Ukraine, to the challenges associated with climate change in Iowa. Before I introduce tonight's speaker, I want to acknowledge the ISU Lectures Program for their role in helping us sponsor and coordinate this event. And I would also like to especially thank everyone from the LES team who was involved with making tonight's lecture possible. This valuable partnership with the Lectures Program heightens our ability to share the college's contributions with the community, and it is greatly appreciated. At this time, it's my pleasure to introduce Elizabeth Betsy Swanner, Associate Professor in the Department of Geological and Atmospheric Sciences. In her research, Professor Swanner investigates midwestern lakes that share common features with ancient Martian lakes and Earth's oceans. In her lecture, she will discuss how some minerals are formed and what they're present in ancient Earth sediments or old Martian lake beds can tell us about past microbial life on Earth or possibly even Mars. Professor Swanner's research has focused on geochemical reactions in water bodies that have little oxygen and are enriched in iron. This environment can be found in a special type of lake that Professor Swanner explores. These lakes do not undergo seasonal mixing and maintain oxygen-free deep waters. With no oxygen, microbes are the dominant life form, and their metabolisms can influence processes from mineral formation to greenhouse gas emissions. LES research truly does transform the world by addressing the challenging issues of our time, and Professor Swanner's work is no exception. She's currently funded by a National Science Foundation Career Award, that's very prestigious, as well as other grants from NSF and NASA. Please join me in welcoming the Fall 2023 Dean's Lecturer, Professor Betsy Swanner. Betsy?
Good evening and hello. It's a real honor to be here, to be invited to be on this prestigious stage, and I'm very honored that there are so many students in this audience. My work is highly interdisciplinary, and I hope that many of you are from around LAS or the university, and I hope that there will be a little bit of something for everyone. Before I get started, I need to make some acknowledgments because I run a research program, and that research program is populated by students who are earning their degrees or postdoctoral researchers who are building their own careers. And so I want to take a minute to acknowledge the team, the GeoBiochem team as we call ourselves, and I'm going to be particularly highlighting work by Nick Lambrecht, Gabby Ledesma, Kaylee Block, Sujata Kam, Michelle Chamberlain, and that was all on my list. Okay. I also need to acknowledge some collaborators who have been heavily involved in the work I'm going to show you tonight that come from international groups, and as well as the funding we receive from the NSF, NASA, and more locally, the Iowa Space Grant Consortium. Finally, work on lakes is necessarily using a public or private resource, and so we gain access to those sites through the generosity of private foundations or through organizations that manage the resources. So I told you I'm going to talk about Earth and Mars, and I'd like to start off with a bit of comparative planetology. So we're going to compare what Earth is like to Mars today. My kids are in elementary school, and one of the things that I learned when I was in elementary school is that Earth is covered in 70% water, something we all know, right? Our atmosphere is composed of gases, but one of the most abundant ones is oxygen, 21%. And although we might argue about it's a little hot in some places or sometimes and a little too cold in other places, in general, the temperatures on Earth are okay for maintaining a liquid water in the oceans and in lakes and other sources of water. And finally, we know for sure, because we're all sitting here tonight, that there's life on Earth. Duh. Okay, so Mars, by comparison, doesn't have really any liquid water. There is some ice on the surface of Mars, and the atmosphere is much thinner than on Earth, and it's composed almost completely by carbon dioxide. And temperatures are generally a lot colder on Earth. They're incompatible with the stability of liquid water, and we don't know for sure if there was or is life on Mars. But despite these differences, Mars and Earth formed from very similar processes. They both accreted or formed from the solar nebula, so gases and dust, that smashed together over time, building larger and larger bodies and finally planets. Once the planets were formed, they developed their own internal structure, taking the heat and energy of all those collisions and developing a core, mantle, and a crust. If we compare the early Earth with early Mars, the situation is still pretty similar. Both of these planets were relatively warm, compatible with having liquid water, and they were relatively wet. Mars might have even had oceans, but certainly it had some large lakes, which I'm going to talk about tonight. Early Earth was much hotter, but it had, in its early history, but it had wet oceans and things like we know today. But if we went back to early Earth, we would have a problem, right? I always think about this when I imagine time traveling. I'd love to go back and see my study sites, but the minute I would take a breath, I would essentially go extinct, right? Because if we think about the early atmosphere, it had a very different composition. Largely methane, that's still the same, but a lot of carbon dioxide, and what's missing? Oxygen. Whereas modern Earth, we know, has 21% oxygen. So this implies that Earth has changed through time. And those changes are really driven in a coupled process referred to as coevolution. So as life began to take hold on Earth and evolve and become all sorts of the diversity that we know today, life and vital processes changed the environment, and as that changed, the Earth system started to change and further constrained the evolution of life. And what I'm showing you on this plot, there's a y-axis with numbers, the percent of oxygen in the atmosphere, and this is a timeline on the x-axis. The earliest period starts about 4.5 billion years ago, and oxygen concentrations in the atmosphere are 1 million times lower than they are today. And that condition persisted for quite a long time, until about 2.5 billion years ago, when there was a biological innovation, the evolution of bacteria called cyanobacteria, that had the ability, or they came up with the ability, to use pigmented molecules to harness sunlight energy and convert water and carbon dioxide to oxygen and sugars to grow their cells. And this started a rise of oxygen in the atmosphere, but as you'll notice, we don't really hit and stabilize at modern concentrations of oxygen until about 2 billion years later. It's a pretty slow process. Meanwhile, what's happening in the rest of the Earth system is it's responding to all of this new oxygen. So oxygen can diffuse quickly when it's in a gaseous phase, but when it's dissolved in water, it's much slower at moving around. And so the early oceans, this is showing sort of a transect of land here in an ocean basin, they were actually filled with iron. And oxygen that was being formed in the top of the ocean due to photosynthesis, where the sunlight was in low concentration. Now I need to tell you something about oxygen and iron. When they're together in water, they sort of destroy each other. They undergo a chemical reaction such that when there's a lot of iron, it's hard to build up a lot of oxygen, and vice versa. And so the transition to the modern ocean that we have, where there's a lot of oxygen and very little iron, actually took even longer than this initial rise of oxygen in the atmosphere. But this is important to get to this last stage, because once we have oxygen fully ventilating the oceans, it allowed for the evolution of organisms like plants and animals and the emergence of life onto land. So one of my questions, given my expertise, has been, why did the oxidation of the atmosphere in the oceans take 2 billion years? It's a really long time. And so as someone who works with microbes and someone who works with chemistry, I took to the lab, and I'm gonna tell you about some experiments that I did when I was a postdoctoral researcher in Germany. I was wearing my lab coat, working in the lab, and I decided to work with a strain of cyanobacteria. So here it is in a fluorescence microscopy image. My students from class will know what I'm talking about. And what I did was expose this cyanobacteria in bottles to different concentrations of iron. And as I ramped up the concentration of iron, the color's changing, right? It's more green, representing better growth, more chlorophyll, this pigment that makes plants and cyanobacteria green. But when there's high concentrations of iron, you really see this rusty color dominating. And so to quantify that something was happening in these experiments, I did what I'll call taking a vital sign of cyanobacteria. So a vital sign for us is our heartbeat or our breathing. And if our breathing rate changes or our heart rate changes, it's maybe signifying that something's going a little wrong. So this measurement over here called yield is a measure of the capacity of cyanobacteria to do photosynthesis. And what I noticed is that as the concentration of iron increased, their vital signs were dropping. So this represented some kind of toxicity due to the presence of iron. We hypothesized that what was happening is that as the oxygen and the iron react, we know that they do that, in the presence of light, it's actually producing free radicals. And free radicals, you probably heard, are bad for you. So they're bad for cyanobacteria too. And in support of this being really the mechanism, we went and measured the amount of free radicals in the cyanobacteria and found that they were indeed elevated when we raised the iron concentrations. So with this, I had this initial idea that cyanobacteria maybe were really slow at producing oxygen on the early Earth because they were living in a toxic environment. But the problem was that I was in a lab and doing all these experiments, and I was sort of ignoring the complexity of the real world, mostly because I didn't have access to it. But that changed when I arrived at Iowa State in 2015. In my first semester, I was introduced to collaborators who introduced me to a number of midwestern lakes, actually 2 midwestern lakes, and then later on we discovered or found more, that have some common features with the early oceans, where we could really go in and look at the complex system, microbes in a complex ecology, a complex geochemistry, and understand what the controls really were on their ability to produce oxygen. So the specific thing about these lakes is there's something called meromictic, and the students from my other class know what this means. It's a Greek term that's referring to a lack of mixing. And so in a normal lake, during the summer, there's sunlight hitting the top and it's warming up, right? And if you've ever swum in a lake in the summer and you put your feet down, they get into colder water. So warmer water is less dense, and so it floats on top of the lake, and the cold water stays in the bottom, and the lake is essentially layered. And normally, this time of year, what happens is temperatures cool down, and the lake water becomes more dense at the top because it's cooled down. And then as the winds come across, which we know they do, right, in Iowa, they mix the lake water, inverting the top water into the bottom. But in a meromictic lake, there's always some amount of water that doesn't get mixed up. And this is due to the action of dissolved ions and salts, because they also increase the density. And so this water down here has temperature making it more dense, but also salts. In the case of meromictic lakes in the Midwest, that ion is actually iron. And so because of the history of how these lakes formed from glaciation, there's a lot of iron around, and there's a lot of iron in the bottom of these lakes. And I'm gonna introduce 3 different lakes: Brownie Lake in Minneapolis, Minnesota, the Canyon Lake in the Upper Peninsula of Michigan, which is by far the most photogenic lake, and Deming Lake, which is in northern Minnesota. So I'm gonna be presenting a lot of plots like this, where on the y-axis, what you're seeing are meters below the surface, and on the x-axis is the amount of something. And I might refer to these as profiles. So in these profiles of Brownie Lake, you can see that there's iron building up in the bottom part of the lake, same as in Deming, where oxygen, the dashed line, is reacting and disappearing away as we get deeper into the lake. So these lakes are stratified by density, but they're also stratified by chemistry. And this chemical layering is what we expect for the early oceans. So we can use these lakes to investigate processes like photosynthesis. Now in these plots, what I'm showing you is the abundance and distribution of photosynthetic organisms, including cyanobacteria in blue, but also some other organisms that are descended essentially from cyanobacteria. And we can't necessarily get rid of them, although they wouldn't have been present on the early Earth. But one of the pervasive observations that we made in all of these lakes is that there's a peak of photosynthetic organisms below the surface, and it's often roughly corresponding to the zone where iron and oxygen are interacting. I'm gonna hone in on Brownie Lake now and spend the rest of the time talking about Brownie, sorry, Canyon. And what we observed in Brownie Lake is when we looked closer in, that there were actually 2 different populations of cyanobacteria. So the cyanobacteria that were sort of at the top part of the peak, where there's maybe a little bit more oxygen, we'll call them high light cyanobacteria, because they lived where there's more light, whereas there's a different type that live in the bottom part of that peak. And much like my early experiments, we've done these yield measurements, or these vital sign measurements for photosynthetic organisms. And what's interesting is that where the organisms are living in the most light, they're the most healthy and fit for photosynthesis. And then this highlight population of cyanobacteria, their vital signs kind of drop. And then we switch over to this lower light adapted cyanobacteria. And although there's more and more iron, and it's maybe getting darker and darker, their vital signs are stable, which indicated to us that there's some process that's maybe mitigating things like toxicity or the other stresses that these organisms might have in their environment. So what we think is going on is something like this. The cyanobacteria that live near the top and are adapted to high light, they can produce a lot of oxygen because there's not a lot of iron around, and it's released into the water and eventually into the atmosphere. But those lower light adapted cyanobacteria are probably not quite as active, and when they release oxygen, it's actually being consumed by other organisms that are living with them. So in the case of Brownie Lake and many of the lakes we investigate, these organisms are called methanotrophs. And that literally means they eat methane. And methanotrophs require oxygen, but they do best when there's only a small amount of oxygen around. And so this is a really nice, it's almost like a symbiosis, except they're not living together. My students actually know this as a syntrophy, where organisms sort of feed together. And in support of this, what we've detected here, based on the types of DNA that we find in the lake, is that we have a higher abundance of methanotrophs. These in the atmosphere, it's less abundant than CO2, which is also kind of trace carbon dioxide. It has about 25 times the warming potential as carbon dioxide. And so when it gets into the atmosphere, it's more of a potent greenhouse gas. It turns out that methane is actually produced during decomposition. So, are you laughing at my video? I'm going to talk about that in a minute. So as cyanobacteria and organisms that are growing in the top of the water column and making these sugars, as they sink, they need to be decomposed. And when there's no oxygen for decomposition, that decomposition has to happen through other pathways. And there's a microbial pathway called methanogenesis, or originating methane, that takes place in systems like lakes. So this does kind of look like a weird Halloween ritual, but it's actually students up at a field station in northern Minnesota where we work. And what they've done is put on waders and stomp around in a shallow part of a lake where there's a lot of organisms growing, and there's a lot of plants and things like that, and there's a lot of dead and dying stuff to decompose. And it's turned into methane gas in the sediments, and the students stomped around, and they had a plastic bin, and they collected all the gas from the sediments, and then they flipped that bin over and lighted it on fire. And so methane is this colorless, odorless gas, natural gas, but it's also combustible. So it turns out that what we found, and this is in line with what people have observed from other meromictic lakes around the world, is that they produce a lot of methane. Because if you think about it, there's never any oxygen introduced into the bottom of the lake where all this stuff is sinking. And so a lot of the decomposition must have to produce methane rather than being decomposed, what we call aerobically, or with oxygen. In support of this, this is data from Brownie Lake showing that we do indeed see elevated concentrations of methane in these different colors are at different times. It's persistent throughout the year. And with our DNA-based techniques, we can also look for this very particular type of microbe called a methanogen. And I just need to mention that methanogens are archaea, which is the third form of life. And if you don't know about them, you should find out. They're really cool. So bacteria, archaea, and then eukaryotes are like all of us, humans and animals and plants. So we wanted to know how much of the decomposition is producing methane, but it's actually like a hard number to measure. And so what we can measure are the distributions and the profiles of all of these chemical species. And then we can use math. And we can use the math to simulate the reactions that the chemical reactions of one, say, oxygen reacting with iron or methane reacting with oxygen, and the ways in which these can be catalyzed by microbes. And we can also use math to simulate the way that these chemical species are moving throughout a lake. And when we do that, we're essentially creating a model. And so the model, in this case, our data are the solid lines, and this is a schematic. I drew this, it's not real. But this is like what we do. Those dashed lines are like our model data, and what we do is we work the model until we get a good fit, and then we can use that model to tell us how much of the decomposition is producing methane. In the case of Brownie Lake, it's about 20%, which is within the range of estimates that have been done on other meromictic lakes, and particularly those meromictic lakes that have a lot of iron. But when we compare that to the modern ocean, which is fully ventilated and for the most part full of oxygen, there's almost no methane being produced in the ocean water column itself. And so what we can take from this is that these meromictic lakes produce a lot of methane, and on the early Earth, when the system was very similar, they might have also been producing a lot of methane in the early oceans. There's one other important observation that we made. This is another fin video. Can anyone guess what it is? I always do this in class. I write what it is, and then I ask my students to guess. So what's going on here is we actually took an infrared camera, and we put it at the bottom of the lake, and what you'll see as this video repeats is that we saw methane bubbling out of the lake, and we actually measured the amount of methane bubbling out of the lake, and it was 10 to 100 times more methane being transported that way than there was sort of diffusing up through the lake water column. And the importance here is that when methane is diffusing up through the water column, it can be eaten, right? It can be eaten by those methanotrophs that are living with the cyanobacteria. Whereas when it bubbles up, it's fast. You know, it's a few seconds, and it's out into the atmosphere. And so this is potentially a very important mechanism for these systems to put greenhouse gases into the atmosphere. So if we take another look at this timeline, in addition to oxygen, we can also think about methane being much more abundant in the early atmosphere than it is today. And likely, a lot of the abundance of methane might have been due to the fact that it would have been more readily produced from the oceans themselves. This is important because there's this problem that people talk about called the faint young sun. So during the early period of the sun's history, it was not as bright, and so there was less radiation hitting the Earth to keep Earth warm and to keep liquid water as liquid. But we know there was liquid water, and so there must have been higher greenhouse gases to account for that. And we think that methane might be an important part of that, and we can now show that this iron-rich ocean may have been producing more methane and releasing it to the atmosphere, and that might be one mechanism for solving this, what's called the faint young sun paradox. So if any of you read the abstract or were paying attention to the introduction, it was promised that I would talk about minerals, and I'm going to do that now. Minerals are inorganic components of rocks that have a defined crystal structure and a defined chemical structure, and because of that, they tend to form under very specific conditions. And so if we see a mineral, we can usually infer something about how it formed or what the conditions were like. And so minerals can actually be very useful for helping us do what we do as geoscientists, which is a lot of detective work. We go back and we look for clues about what happened. So in order to start talking about minerals, we need to do a little bit more chemistry. So I'm adding some chemical notation here for oxygen and iron, and I'm also going to introduce another element called manganese. Manganese is different than magnesium, by the way. A lot of people get that confused. But I tend to think of manganese as like iron's little sister. Iron is one of the most abundant elements on Earth. Manganese, not so much. But like a little sister, it behaves a lot like iron in that it reacts with oxygen, but it needs a little bit more oxygen around to do that reaction. And so if we think about the profiles of manganese and iron in a meromictic lake, it might look something like this. So let's put together what we've built so far. we have, in a system like this, these highlight cyanobacteria that are producing oxygen, and they're taking up CO2 from the lake and converting it into sugar, carbon dioxide. And when that happens, they sort of perturb the chemical equilibrium of carbon dioxide in favor of this form of carbon dioxide called the carbonate ion, which then can react with calcium that happens to be there and form a mineral called calcite. And so if we form a mineral in water, it's gonna sink, right? Okay, I'm seeing Amy nodding, yes. So what happens is it starts to sink into the next zone where there's a lot of manganese, and there's a little bit of oxygen being produced by these low-light cyanobacteria, and it's being reacted by those methanotropes with the methane, and they're converting it into more carbonate. But now, because there's manganese around, the manganese and the carbonate precipitate, and they actually form a rim, because it's just easier to make a new mineral on something else. So they form a rim on the original calcite. And so what deposits in a system like this, Brownie Lake, and this is happening in Brownie Lake, is that when the minerals get to the sediments, there's a grain of calcite, and it's coated by manganese carbonate. However, there's another way that we can make manganese carbonate. So when there's a lot of oxygen being produced, and it's in a zone where there's manganese, as I already told you, they're gonna react. And they react directly to form this mineral called manganese oxide. Manganese oxide, again, is heavy and it sinks. But before it does that, its surface is very chemically reactive. It's like a sponge, and it's a sponge for other metals. And I'm abbreviating them here, things like cobalt, barium, nickel, zinc, strontium. And so this mineral becomes coated in metals. And then it deposits to sediments, and all of that sugar and decaying biomass deposits as well, and new microbes in the sediment can actually react those two together to produce manganese carbonate by a different pathway. But in the process, we form these manganese carbonates that don't have the calcite rim, or the calcite center, right? And the sediment should also be enriched in metals, because once you get something into a sediment, it's kind of stuck there. So we're gonna take this to Mars, finally. I promised Mars, I'm bringing you there. Mars had water in the past. We already talked about this. So about 3.5 billion years ago, there were lakes on Mars. And this lake is actually in a former impact crater where there's a meteorite impact, and it filled with water. And it turns out, well, we've sent a rover there. NASA sent a rover there called Curiosity. And it turns out that in Curiosity's travels, it's landed on the base of the lake, former lake bed, and it's gone up this mountain that happens to be in the middle. And at each of these spots that's circled, what it's found are elevated concentrations of manganese. And so elevated because I told you manganese isn't very abundant on Earth. It's not very abundant on Mars either, about equivalent. And so most of the sediments that Curiosity has encountered, lake sediments, don't have more manganese than the rest of the Mars material. But there's these few spots where it's really, really enriched. And so we're very curious, since this is happening in a lake, whether or not we can constrain with the minerals that are present what the type of setting was and what the processes were. There's a problem though. Curiosity has a specific payload of instruments, and those instruments are good at certain things, and there's one that's good for determining mineralogy. But it really only works well for minerals that are in high abundance, or for minerals that have a really good, clean crystal structure. And it turns out that sometimes manganese and iron minerals don't. They're either in low abundance, or they don't have a good crystal structure. And so there's not been a really easy way to determine what minerals are present that are forming these manganese enrichments. Luckily, Curiosity has other tools, including on the mast, this instrument called ChemCam. ChemCam has what's called a laser-induced breakdown spectroscopy. And you guys know the store ray gun, right? This is like actually a ray gun. Like it pew, 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 points a laser at rocks, zaps them, and then it can detect what elements are present in the rock that it just sapped. But LIBS is only really telling us which elements are there and not the minerals. And we need to know which minerals were there in order to tell the story of how these manganese enrichments occurred and what the past environment was like. So in our research program, in our research group, we hypothesized that the elements actually indicate the manganese minerals and the conditions. And it goes something like this, and this is sort of a review because we've already been over this, that we can form in a chemically stratified lake system calcium carbonate, which then can accumulate a rim of manganese carbonate. And in that case, we'd expect calcium and carbon and manganese. Oxygen's everywhere, so it's not very diagnostic, and I'm going to kind of ignore it. Whereas if manganese formed directly from reaction with oxygen and then was transformed to a manganese carbonate, we would not expect the calcium carbonates, and we would also expect elevated amounts of trace elements. And there's one other potential pathway, which is that those manganese oxide minerals form and they never get transformed. And in that case, you'd also expect high concentrations of metals, but no carbon. And importantly, these two scenarios over here involved higher concentrations of oxygen, remember? Because manganese is the little sister, it needs higher concentrations to react than iron. We don't know if there was oxygen on Mars or oxygen that was very abundant on early Mars, but there is this other compound called perchlorate that's been detected in the Martian regolith, or the surface, and that can also oxidize manganese. So if we find these types of minerals, or these types of elements inferring these types of pathways, it would infer that there were high concentrations of either oxygen or perchlorate. Whereas if we find a system more like this, it would implicate lower oxygen or perchlorate concentrations. So what we did then was to look across all our collections of minerals, both from lakes and from other earth materials where these processes might be happening, and we took manganese carbonates from lake sediments and manganese carbonates from old marine sediments that had since turned into rocks, and we accessed through drill core libraries. We also took samples of these manganese oxides that form in lakes and never transform and are enriched in metals, as well as there's samples and data from these types of deposits. They cover vast regions of the ocean. So we took all those to Los Alamos National Lab, and it turns out that the people there made ChemCam, and they have an extra one. And it's set up exactly like the conditions on Mars. And so we could analyze our samples in there, so we can compare our data that we get on these Earth materials to the data that's coming back from Gale Crater. So what we found is that elements can indeed distinguish Earth's manganese minerals. So what I'm showing here now is down here is low amounts of nickel, and over here is low amounts of zinc, getting higher and higher. And all of these diamonds are manganese carbonates. And they have overall lower abundances of zinc and nickel, the trace elements, because we wouldn't expect them to be present during the mineral's formation. Whereas the lake manganese oxides have high concentrations of zinc, but the marine manganese oxides have high concentrations of zinc and nickel. So not only can we distinguish the mineralogy, we can also distinguish the setting on Earth. So what we've done is to take these data from marine manganese oxides and lake manganese oxides and manganese carbonates and compare it to the data sets that have come back from Gale Crater. What I'm showing you now is data for strontium. And what you might be saying is, well, first, all of those dots are individual data points, and the boxes are like average. You could think of them as being averages. Okay, so the data coming back from Gale Crater goes through all of our data ranges, right? But I think that's actually consistent with what's been found on Mars in Gale Crater from other studies. For instance, one study that I was recently a part of found these manganese nodules at higher elevations in the lake and inferred that they might be something like a manganese carbonate or a manganese phosphate. Whereas earlier, when Curiosity first landed on the lake bed, the inference from multiple lines of evidence was that manganese oxides were what was hosting the minerals in the elements. So with that, I want to sort of wrap up, but I want to give you a sense that as a geoscientist, if we're interested in past processes, we can do a couple of things. We can send a detective to the scene of the crime to collect evidence. In the case, it's Curiosity, or maybe we're going to early Earth and picking, or not to early Earth, we're going to pick out early Earth rocks that might still exist on the planet. But the problem is that the scene of the crime has been sitting there for a long time, in the case of Mars, and processes have overprinted it. Or in the case of Earth, we have this coevolution of life in Earth that has dramatically changed the scene of the crime. However, even if we can't find direct evidence of every type of evidence we need at the scene of the crime, we can use these lakes to try to fill the gaps. So I've shown you tonight that the lakes have helped us to understand something about how greenhouse gases might have been produced from the early oceans, how minerals might have been formed that can be diagnostic of the conditions in the lake, and how oxygen production might have been regulated in these systems. And so with that, I'm going to wrap it up.
Well, thank you very much, Betsy, for this tour of Mars and northern Minnesota lake and oceans and all of that. It was very fascinating. So I'd like to open the floor for a few questions. There are microphones on each side of the room, so if you would please use them so people who are watching us online can also follow the questioning.
You talk about manganese coming in different forms, tying up with carbon. I would think iron would tie up with carbon as well, and that iron isn't just iron, that there are hundreds of kinds of ways you might find iron.
Yeah, that's right. And actually, there's a lot of analogous minerals between manganese and iron. So iron makes iron oxides. It makes iron carbonates, just like manganese. And so some of those minerals have been detected, for instance, on Gale Crater or from the early Earth. But sometimes they may be transformed into a slightly more stable version of a mineral. And so we still need to basically do this kind of work with iron as well as manganese to understand what the conditions were like that actually formed them. Good question.
Yeah, no, wonderful talk. When you looked at the lakes on the Earth, you talked about bacteria being involved in those processes. So sticking your neck out, what might this tell us about the possibility of life on early Mars?
Yeah, so it's a great question, and it's a natural question. My work, I think, when I'm talking about Mars, and we're really looking for the compatibility of the system with life, because it's actually really tricky to detect life on a planet that if you don't know what's there. You need to develop criteria for what life is, and that's something we actively argue about as scientists. And so my work really is saying these are the possible pathways which this could form on Earth. Some of them are biologically mediated, and in some cases there are analogous processes that are carried out completely abiotically that are plausible for a Mars that doesn't have any life at all.
Mm-hmm. So I was really fascinated that the oceans are actually completely ventilated. I think that's the term that you use, but these lakes still have some of these ancient features. Can you talk a little bit about what's so unique about the lakes? Is it their age? Is it their depth? What makes them so different?
Yeah, that's a great question. So and I glossed over this because I didn't have time, but the lakes are meromictic because it's something about their shape and their size. And so they tend to, I always use this analogy of if you have like a tall cup of coffee and it's hot and you blow across the surface, it's a pretty ineffective way to cool down the tall cup of coffee. But if you have a soup bowl and you blow across it, it's much more effective, right? It mixes more readily. And so these lakes are sort of tall and small. And so that's one of the reasons why they don't mix. Whereas the oceans are undergoing mixing constantly, and it's a much larger and longer scale process. And that, over time, has sort of kept pace with the processes that are consuming oxygen.
Does Ada Hayden turn over? What kind of a lake is it?
I think so, but I've never measured it. My understanding is it formed after quarrying activities, so. I'd love to hear questions from students, just a little bit of pressure.
If there was one tool you could stick on the Mars rover today to best benefit your work, what would it be?
Oh, one tool on the Martian rover. So I would have to say that for the types of minerals that I'm interested in, I actually often go to a national lab that has something called a synchrotron, which is essentially a giant, like half a kilometer diameter thing where electrons are spinning around at the speed of light and giving off X-rays that are very powerful and can actually look at the more detailed chemical structure of these minerals. That's pretty hard to put on a rover, but I think there are people who are trying to scale down a similar type of analytical toolkit for future rovers.
Far for me. What % of our lakes are don't get mixed in the Midwest?
The truth is we don't know, but I would say it's probably around 1%. So we actually did an analysis where, because something like the lake shape is really important, and the lake shape is something that is typically measured by, for instance, in Minnesota, the Department of Natural Resources. And so we took all that data and we calculated that about, I think it was 1.7 or 1.8% of the lakes that Minnesota had data for have the right shape to be meromictic. But the issue is that sometimes, if there's not enough of a salt component or some other component, it might still mix. And so it really requires this detailed field work. And that's one of the projects we have ongoing in the lab, is to try to find more of these lakes. So some of the students are here. We're actually going out to new lakes this semester and collecting the data that we really need to see that it's not mixing in the key moments when the seasons transition. So that's very detailed work that we're doing. Yeah.
So in learning more about aquatic sciences and fishery stuff with my major, I've learned a lot about the thermocline, it's called, the layer in lakes usually where
Yes.
the levels of oxygen can't sustain certain forms of life.
Mm-hmm.
If it does relate, how does it relate to the type of layering that you were talking about?
That's a great question. So in addition to layers of density and layers of chemistry, there's also layers of other physical properties. And the thermocline is where you have high temperatures and they transition to lower temperatures across a certain boundary. The thermocline is often corresponding to where we see this high amount of chlorophyll and these photosynthetic organisms, because what they're doing is they're balancing the availability of light and the warmer conditions at the top. But I didn't really mention it, but there's a lot more nutrients in the bottom of these lakes because of decomposition is actually releasing those nutrients. And so they're sort of balancing it and usually living right at the thermocline.
So I have zero geo background. I have a very naive question. So when you do those measurements, like, how do you, I don't even know how to ask this question, I guess, just naively,
Yeah.
how do you go to
How do we
the
get the
steps
samples?
to
Yeah.
get the samples, and how do you not disturb the water to, like, introduce external termination
Yeah,
of the
that's a
the
great
fluid?
question. Right, because we're looking at these very fine gradations, and by introducing something, we might mix it up. So what we actually do is we sit on a boat, and I'm like looking at the student over there who's in my class, and like we just did this a couple weeks ago. We sit on a boat. It's usually a small boat because they're small lakes, like a canoe, and we typically drop a pump down. So there's something that goes down into the water, and we tie to it the sensors that tell us the measurements of things like temperature and oxygen, pH, a few other parameters. And we use that to find the layer that we're interested in, and then we have the controller for the pump at the top with a battery, and we just bring the water up to the top of the lake, and we fill all our bottles and everything that we want to go back to the lab and make measurements on. And we're making the measurements, or we can monitor things like the chemical conditions down there as we're sampling, and it doesn't perturb those things. Yeah.
Just a quick follow-up. So I guess, so at the moment, like when you sampled it, you measure some of the readings, and then you take back to lab, do you try to replicate, for example, the pressure, temperature, that kind of stuff?
Oh, that's a great question. So we do a lot of, your question was, do we try to, when we make our measurements, account for things like temperature and pressure that are now different? We do a lot of tricks to maintain or preserve the sample. And so sometimes we're actually sampling right into, say, an evacuated vial. Evacuated means it's under a vacuum, and there's no oxygen in there. We're putting things on ice. We're adding acid to keep something stable. And so there's a lot of those steps that happen to make sure the samples, when we analyze them, are really representing the data that's in the environment.
Thank you so much.
Great question.
Hi, you mentioned earlier that the shape of the lakes here affects the mixing and how you gather the data. How does that compare to the shape of the lakes on Mars, and do you take that into consideration when you gather data?
Yeah, it's a great question. And so the lakes that I'm working on in the Midwest tend to be very small. They're actually formed through, most of them are formed through glacial processes, and they're actually glacial kettles. They formed when a block of ice came off the retreating glacier and then sort of got covered in debris and then melted, sometimes over thousands of years, and formed this relatively deep lake. But that's not at all how the lakes on Mars formed. But there are meromictic lakes on Earth that are huge. They just happen to be in tropical locations. So the African Rift Valley is a great example where there are really famous meromictic lakes that are gigantic. So I think that the lake that was in Gale Crater or in Jezero Crater, where the Perseverance rover is, were probably more like these tropical lakes. And in that case, because it's warm year-round, you never have the density of the top water changing, and so they don't mix because there's always a thermocline.
So Brownie Lake and the lake in the Upper Peninsula are both in the iron range. What effect, if any, did the presence of iron have on your analysis?
Yeah, so Canyon Lake is in the iron range. It's not next to any iron deposits. So it's actually in a rock called Archean gneiss, which has iron, but it's not really that much different from any other type of rock. I do think, and we're doing other studies on this, that the source of the iron to these lakes is really coming from the rocks and the material that it's in. And in the case of glaciation, what we think is really important is that glaciers actually are really good at chewing up rocks into smaller and smaller pieces. And some of that iron can start to react and form these certain minerals that then, when they're buried and water is moving through, the groundwater is moving through, can become dissolved, and that groundwater actually moves into the lake and brings the iron from the surrounding aquifer. So there are lakes in the Iron Range that are pits from mining, and some of them are meromictic. They tend to be more, sometimes they can be more acidic, so I tend to not work on those systems because they don't have quite the right profiles for me. It's a great question.
Well, great questions. Thank you all very much for asking so many questions. That was really interesting just to hear that discussion. Once again, please help me in thanking Betsy for this very informative and exciting lecture. And I would like to invite all of you to stay for a reception. We have some refreshments at the back there, so you can continue to talk to Betsy and maybe ask all the questions you didn't ask right now. And I also invite you to explore research and other news on the college website, which is las.iastate.edu. And once again, thank you all for coming. Have a great evening. And once again, thank you, Betsy, for a great lecture.